Good morning. This is Debbie Hart, the President and CEO of BioNJ, and thank you for joining us today for our first of two COVID-19 rapid fire research showcases. To everyone on today's webinar, I hope you and your colleagues and your families are staying safe and well in this really difficult time. And during this unprecedented time, we are so proud of the unwavering commitment and tireless work being done by the life sciences industry. It's really extraordinary. Today is a reflection of BioNJ's commitment to trying to make a difference in COVID-19. More importantly, though, we have today 10 companies who have looked in their portfolios, found something that they hoped or thought could work on COVID-19, and they will literally be spending, pouring their hearts and their souls and spending hundreds of millions of dollars to trying to make a difference. So we applaud them. We will hear from 10 companies today, another 15 on June 16th. We're hoping today the format is really intended to encourage new connections, partnerships, and collaborations, because as we say at BioNJ, patients can't wait, so we need to get this done quickly. From vaccines, cell and gene therapy, treatments, and therapeutics, our presenters will highlight the important science coming from New Jersey and, in fact, globally. So I'd like to thank our supporting sponsors who you'll be seeing on the screen right there. And also they are our supporting partners and also our sponsors for making today's presentation and our series possible. Before we get started, just a very few logistical reminders about the program. So this is a listen-only webinar. However, you can ask questions. You'll see a dialog box on your screen. I would ask you, because we have not a lot of time, lots of speakers, if you intend to direct a question to the group, I would ask you to please direct it to an individual person or persons. We have a good sized group on the line. We'll try to get to as many questions as possible in the time we have allowed. And if we don't get to your question, we will follow up with you after the webinar. There will be a recording of this webinar on the BioNJ website tomorrow at bionj.org. So now the person who's really been tracking the commitment of the industry globally, it's really been extraordinary. She'll, I'm sure, be able to share some information on the pace that she's been on, because the pace that you've all been on trying to make a difference. So I'd like to welcome Vice President of Infectious Diseases and Diagnostics Policy for the Biotechnology Innovation Organization, better known as BIO, our friend Phyllis Arthur. Phyllis, please take it away.
Hi, Debbie. Thank you so much for inviting me to join. And hello, everyone. Again, I hope you're doing well. And certainly, I agree with Debbie that companies are working at an extraordinary pace to bring really important COVID-19 products to all the people in the United States and around the world. So I'm going to share with you information from a tracker built by our fantastic industry analysis team at Bio that looks at all the details of what's being produced, researched, and investigated for COVID-19. So I'll share with you some information from the tracker, as well as just a quick slide on some of the challenges that all of our companies face in the biologics space. Our approach actually is to take information from a couple of other trackers and then, of course, apply some of the great scientific understanding of our people at Bio to make a tracker that's more detailed. So it has a lot of information, not just on the companies, but who they're partnering with, who's funding the research, where they are in phase. And I put here the website where you can find the tracker yourselves and download data, bio.org/COVID pipeline tracker. And the website comes up again at the end. So I think one of the things that has been so amazing is if you think about when the genetic sequence data for SARS-CoV-2 was first made available in publications in December, January, from then, companies started making products or investigating whether their products would work. And we had some vaccines and even some antibodies in animal trials very, very quickly. And right now we're tracking 550 unique development programs in 20 weeks, which is an incredible amount of scientific investigation. And you see, we literally add to this table every week. There's about 30 new programs just from last week's tracker. What's really important to note is 70% of the products, the drugs and vaccines that are being investigated are actually being investigated by small companies. A lot of really novel science is being brought to bear. And you'll see that's across antivirals, treatments, and vaccines that small companies are doing a lot of the research. And I think it's actually very important to say, and I'm very proud to say, that a great deal of that work is happening in the United States. A lot of US companies or companies with very strong US bases of research, development, and manufacturing are doing the investigation of these important products. So it just shows the strength of the biotechnology industry in our country, the life sciences industry across the board. And I'll say, in my work at Bio, knowing that we work across the life sciences, that's human health, animal health, and industrial and environmental, many of the companies that have expertise in animal health are offering up their capacity and capabilities to companies in the human health side, because we're really going to need to go quickly when we're going to have to leverage every manufacturing and scientific endeavor that we can to respond to this pandemic. So the work across the life sciences in the United States is also a testament to the commitment of our industry. So we're tracking this in three pipelines: vaccines, obviously, antivirals and antibodies, which are really meant to clear the virus, get rid of the virus, and treatments for COVID-19 illness. And this is obviously a very deep pool because, as all of you are very aware, the disease itself is extremely complex. It's a multi-system, multi-organ disease when you have severe cases. And so the need for things to treat the various outcomes of being infected with SARS-CoV-2 are also leading to a lot of innovation. So here's a slide that really looks at the clinical stage. Of the 550 products, note how many are actually in human trials. This is extremely important because there is a lot of work going on to take either products that are already licensed and that may be indicated for another disease or indicated to treat one of the important symptoms of COVID-19 illness, or to take antivirals that were being researched for another purpose and try those different products in COVID-19 patients. So you see a lot of different products already in human trials, many being repurposed or redirected. And then you see mainly novel vaccines being applied to try to prevent COVID-19 in the future. A lot of different modalities. I think what's very exciting is that 60% of the products that are being tracked are actually not your classic pill or capsule, your small molecule. They actually leverage important biologics technology. So very different approaches across biologics, cell therapies, antibodies, plasma, RNA, and then many different approaches to vaccine development as well. I think, again, that top part on treatments reflects what I said before, which is given the many complications of having a COVID-19 infection, there are a lot of different approaches to treating the patient. You saw a lot of work going on, of course, in the beginning to handle the massive inflammation that was happening in the lungs, one of the things that was leading patients to be on a ventilator. Then you see a lot of different work going on to try to stimulate the immune system on its own to clear the virus. And as we started to see problems like blood clots, new products entering the market that could help with the anticoagulation, things that could help with cardiovascular and renal function. So different products, many of these products being repurposed from existing indications in order to see if they can help get patients well who are in the hospital with severe COVID-19 infections. And then you see a host of different approaches to antivirals in order to try to clear the virus out of the body. And they have very different approaches. Some are meant to try to keep the virus from replicating, keep it from transmitting, keep it from getting into the cell altogether. And these different approaches are going into human trials in the summer. And then, of course, we have a host of different vaccine options that have very different ways of generating neutralizing antibodies, and many of them are going into trials very soon or have started to have their first phase 1 data. So this is our vaccine pipeline watch list. You're seeing a lot in the news about vaccines. There's a host of vaccines being developed, and we're seeing, I think, quite pleasingly, a lot of vaccines in the United States that are about to go into human trials. So even those ones that are listed on the preclinical side are going to be going into human trials most likely this summer, and several vaccines that already are starting to have their first phase 1 data or are planning to do their studies in phase 1 and phase 2. It's interesting, I say, when we make a slide on what's happening with these products, invariably within 24 hours, it's wrong. So Novavax at the bottom there just got a contract with the Department of Defense. So we tried to show where the different funding is happening from the federal government. And in 24 hours, I'm already wrong. There's another product that's gotten federal government partnered funds. So what are the challenges? The challenges actually across the biologics industry are somewhat the same, and they have to be solved obviously slightly differently. But very importantly, a lot of scientific challenges when you're trying to do R&D for a disease that you're still understanding while you're doing the R&D, that is complicated. So companies are trying to understand what's happening with the disease, what are the severe outcomes, what's the incidence of those. Understanding what populations are at highest risk. In the beginning, we were all focused on seniors and the Medicare population. And now with the Kawasaki-like syndrome in children, we need to understand better what the potential attack rate is of children who are mainly asymptomatic, but there's this rare but very important consequence. And the shifting epidemiology as we go through reopening our states. Will we see a spike? It's unclear. How many people are already infected? These kinds of issues will affect the ability to do the research. And then, of course, we're trying to manufacture a product in record time. So companies are looking at scale up. They're trying to find access to capacity in order to make much more product than normal. Putting the product into vials and syringes and packaging it is turning out to be one of the bottlenecks everyone's trying to solve. And of course, we're going to need glass vials and syringes and swabs and all the things that go with giving biologic products and vaccines. And last, it's extremely important to our industry that all this work to add COVID-19 to our manufacturing and research doesn't harm patients who currently are receiving products that are made by our industry. So companies and the government are working together to make sure, although we need to manufacture COVID-19 products, that we don't disadvantage products that patients need every single day. So these are some of the things you can see in the tracker. It's global, so you can look in, you can get into it. You can actually click on any one of the pieces of the pie and just look at all the data for vaccines or antivirals only, or you can look at all of the data. You can see the different products in development, how they're partnered, who's funding them. You can also look at where different research is going on around the globe. So please feel free to use the tracker for your own information. And we look forward to hearing from the companies presenting today. Thank you very much for your time.
Thank you so much, Phyllis.
My pleasure.
It's just incredible. First of all, the yeoman's work that you're doing and the work and commitment by the industry is just obviously unprecedented and just so much needed. And I was just thinking, you know, being wrong in this case is actually a good problem to have, right? Actually changing your data every day is a good problem to have. So thank you so much for this work. It's really important.
Thank you.
Going to hear from some of the companies that you were just talking about. It's my pleasure to introduce our first company presenter. So I'd like to welcome the CEO and founder of Allied Innovation Systems, Dr. Simon Bystriak. Welcome, Simon.
Good morning, everyone. We are a small research and development company, and we are developing new technologies in the field of clinical diagnostics. And our research has been recognized by NIH. We received several SBIR grants from NIH for the development of our technologies. And as you all know, the impact of COVID-19 outbreak could have been mitigated if weak and inexpensive tests to detect carriers of the disease were readily available. And the gold standard right now are nucleic acid tests, but they're complicated. They have serious disadvantages like limited clinical sensitivity, which is around 70–90%. These tests require skilled labor and sophisticated instrumentation. That's why they are very expensive, and probably they are not very suitable for mass testing of general population. So the alternative to nucleic acid tests could be immunoassay-based detection of SARS-2 antigen or antibodies. But these tests are also not very sensitive. For example, ELISA, which is widely used, it's kind of an industry standard. The sensitivity of this test is inadequate. So we developed a so-called AmpliFlux technology for increasing the sensitivity of the conventional ELISA. And we have shown that our technology allows to lower the limit of detection by dozens time. For example, we show that the analytical sensitivity of SARS antigen and antibody detection are approximately 30 times higher than those for conventional ELISA. So our technology is very simple, is based on the conventional ELISA. It doesn't require additional equipment except a small illumination box. And right now we are looking for more investment to commercialize this technology. We developed another technology which is for rapid detection of antibody detection, but this is probably the topic of the next presentation right now. And as I said, we are looking for additional investment. We will be happy to collaborate with anybody who is interested in our technologies. Thank you so much for your attention.
Thank you so much, Simon. And now we'd like to hear from the CEO of BioAegis Therapeutics, Dr. Susan Levinson. Welcome, Susan.
Good morning, everyone, and thank you to BioNJ for this opportunity to present this morning. I'm here today to share how recombinant human plasma gelsalin is expected to address the COVID-19 crisis. But BioAegis is about more than COVID-19. Our mission is to address medical issues previously thought to be out of reach. That is to prevent the damage caused by excess inflammation without suppressing the immune system. We expect our approach will save lives and reduce the morbidity of chronic disease. Our passionate leadership team has over 60 years of pharmaceutical experience, and we're developing technology licensed from the Harvard University hospitals. Recombinant gelsalin is expected to be a life-saving prevention of organ failure in COVID-19, as well as other severe pneumonias, whether the cause is bacterial or viral. It is a key component of our immune system. It regulates inflammation and it fights infection. So why are we so confident that recombinant gelsalin can save COVID-19 patients? Gelsalin is a normal protein circulating in our blood at high levels and a master regulator of inflammation. It boosts the ability of macrophages to take up and kill bacteria of all the types that we have tested. It promotes the resolution of excess inflammation and protects organs, especially the lungs. But this protein becomes severely depleted in inflammation, injury, and infection, including in COVID-19 patients, which is currently being studied at NIH. Patients with comorbidities would have depressed levels even before infection. Supplementation is expected to prevent or reduce the cytokine storm in patients with severe COVID-19. Organ protection and improved survival has been demonstrated in more than 20 animal models of diverse diseases, including models of cytokine storm. So what's next? We're excited to share that we expect to initiate our randomized, placebo-controlled, double-blind phase 2 study in severe COVID-19 patients this month. The study follows a phase 1b/2a study, which was completed last year in hospitalized pneumonia patients, which reported no safety signals, even at doses of gelsalin that raised levels well above normal. While many companies are pursuing antivirals and vaccines for the pandemic, there are few that are addressing the severe cytokine storm that are not also immunosuppressive. Recombinant human plasma gelsalin has the unique property that it is not pathogen specific, and thus will be a solution not just for COVID-19, but for future threats which emerge as well. Thank you for your attention and your interest. Please visit our website and follow us as we move forward.
Thank you, Susan, so much. And next, we'd like to move and welcome Dr. David Mazzo, the President and CEO of Caladrius. Welcome, David.
Good morning, everyone. It's a real pleasure to participate in this panel this morning and to introduce to you CLBS 119, Caladrius's program for the treatment of COVID-19 induced lung damage. I think we all know that the human coronavirus disease, or COVID-19, is caused by a SARS-type virus, the SARS-CoV-2. And preliminary published evidence indicates that this is in fact a disease that permeates the microcirculation, causing damage throughout the cardiovascular system, with specific emphasis, at least in the early diagnoses, in the lung. Evidence from autopsies from patients who have succumbed to the disease show three common and pervasive characteristics of the infection. One, there is a severe endothelial injury, which is due to intracellular penetration of the virus into the endothelium. Two, there's widespread vascular thrombosis with microangiography and alveolar capillary. And three, there's evidence of angiogenesis, which is the body's attempt to repair damaged lungs disease. Now, prior data from a previous SARS epidemic also suggests that CD34 positive cells, the lung progenitors, could be the target of this particular virus, and that destruction of those progenitors could account for the inability of many patients to recover from the severe lung damage provoked by the disease. Our solution is in fact CLBS 119, a proprietary formulation of autologous CD34 positive cells that has been shown in preclinical and clinical studies to induce the formation of new capillaries to provoke angiogenesis of the microvasculature in tissues where the microcirculation has been compromised. Our program has an active IND, which was authorized under the Accelerated Access Program at the FDA, and we're in the initial planning stages about to start a phase 2 proof of concept study, which will focus on two cohorts of patients. One, with which many of you are familiar, those patients who have had the disease, have been added to respirators for assistance, have been successfully extubated, and still have damage. And then the second is that you have patients who are now called, which in some respects is an oxymoron in the medical field, stable respiratory patients, patients who have cleared the virus, have no other infection systems, but simply can't be extubated because they can't oxygenate properly. We'll be studying a single intravenous in solution of autologous CD34 cells to look at how these cells repair the endothelium, and also, of course, managing safety, although I will mention that throughout the more than 1,000 patients who have been exposed to our cells, we've never reported a cell-induced adverse event. We're looking forward to starting our trial in the third quarter of this year. I thank you for your attention.
Thank you so much, David. And now I'd like to welcome BioNJ board member and CEO of Chromacell, Christian Coughley. Welcome, Christian.
Good morning. Really excited to be here today. I'm delighted to share with the community here a research technology that might be able to overcome a key hurdle in the discovery and development of novel vaccines and therapeutics. Cell lines stably expressing proteins are a key research tool in discovery and development of novel therapeutics and vaccines in general. A challenge in COVID-19 is that these cell lines are cytotoxic, the proteins are cytotoxic, which virtually makes it impossible to create stable cell lines needed for development under those circumstances when you're using traditional methods. Our Chromwork technology is poised and designed to overcome exactly that challenge, creating stable cell lines that express the COVID-19 protein in a way that it overcomes cytotoxicity, thereby providing a really valuable tool for the development of novel vaccines and therapeutics. What we propose to the community is creating a couple of cell lines expressing combinations, including new strains of COVID-19 proteins, for furthering the development of therapeutics and vaccine space. With that in mind, we have used the technology to express the epithelial sodium channel. That is a target that's validated for control of fluids in lungs. This is particularly important in the COVID-19 space because fluid buildup in lungs by patients affected by COVID-19 virus is critical and oftentimes lethal. Our research has produced a couple of compounds, both novel and already FDA approved, that modulate the ENaC channel. This could be a breakthrough in the sense that we now have compounds, including FDA approved compounds, that can stop the excessive fluid in lungs by COVID-19 patients. We would love to partner this program with parties that can help us advance in clinical trials to see whether the compounds that we have identified in vitro and show promise in patients, hopefully providing novel therapeutics for COVID-19 patients on a very rapid basis. Thanks for your attention today. I would love to be in touch with anyone who is interested to either collaborate with the cell line technology that we have or in the ENaC space with the novel therapeutics that we want to develop clinically. Thank you.
Thank you so much, Christian. And now, welcome to President and CEO of Everest Bio, Dr. Lilian Chang.
Hi, thank you, Debbie and BioNJ. I'm Lilian Chang, CEO of Everest Bio. So I think the COVID-19 pandemic makes it clear that a radically new approach is needed to address viral infection. Our industry has risen to the occasion, but the problem with our current technologies is they cannot provide us a solution for a new virus in time to flatten the curve. We're learning the hard way that it takes at least 12 months to develop and widely distribute a new vaccine or antibody. As for antiviral drugs, these are relegated to repurposing old drugs like hydroxychloroquine with different outcomes. And so as such, we will not be able to avoid the tremendous health threat and economic cost of another COVID-19-like outbreak. Here at Everest Bio, we're developing next generation antivirals. These engage the biology and the natural defenses of the infected cell so that our antivirals are simultaneously active against very different virus types. In fact, our drugs are designed to be oral, shelf-stable, easily manufactured, and ready to use so that they will be available when it makes a difference to flatten the curve at the onset of the next pandemic. In fact, at Everest, we have 4 distinct product lines, each addressing a very different infection condition. We are targeting the dozens or so viruses that can cause respiratory infections. We have compounds that are simultaneously active against hepatitis B virus and hepatitis C virus. We have a program to target transplant infections as well as emerging highly pandemic infections. If we go to the next slide, please, you'll find our contact information at the bottom of the slide. I'd like to share some data with you. This is data from our Everest lead drug. We've tested against over a dozen viruses representing different viral families that include RNA and DNA viruses, enveloped and unenveloped. If you compare column 3 to column 4, we're looking at the 50% effective dose of the Everest drug against the standard of care. This is known as the EC50. And in almost every case, the Everest drug is lower or better than the standard of care. Here's the kicker. In column 5, each of these standard of care comparators is a different virus-targeted antiviral that is not expected to work on the other viruses in the table. And so I want you to remember our company, so I'm going to spell it for you. It is EVRYS, not just one virus, every virus. Thank you.
Thank you so much, Lilian. A reminder for everyone that the Phyllis and the panelists will be more than happy to answer your questions. You can enter them in the chat box. Meanwhile, next up is the CEO for Maven Diagnostics, an IMS company, Richard Einhorn. Welcome, Richard.
Thank you, Debbie. Maven Diagnostics is not a new name. It is a new name in New Jersey, but our experience is long. Maven is a spinoff from Intelligent Materials, which has developed and commercialized diagnostic tests for tropical diseases for more than 10 years. We are located in the SRI complex in Princeton. We are focused on developing and commercializing what we call high impact tests. That is, tests that provide fast results so that actions can be taken quickly, tests that are sensitive to the disease that it's being targeted for, so medical professionals can diagnose accurately, and simple to administer. So the test is accessible to low resource areas or point of care applications like Minute Clinic and nursing homes, and so you don't have to have a stick stuck halfway through your brain. These high impact tests, they should be able to provide trusted data for which people to make decisions on. Maven is working to develop solutions that combine the benefits of the two primary solutions that exist today. We all are familiar with PCR, the high diagnostic value molecular tests. They're very good tests. They have very high sensitivity at very low virus level, but these results can take several hours and sometimes even several days. The other group of tests are more accessible to the public, and those are serological antibody tests. And their top benefit are that they're simple, they're fast, they're inexpensive, but the results have limited value due to high false negatives. In fact, so much so that the FDA had to take more than 20 off the market in the last few weeks because they just did not perform as expected. We believe there was a better option to develop a more useful test that had higher impact. Our CoV-2 Intelligent LFA, lateral flow assay kit, achieves virus limits of detection around 100 SARS-CoV-2 virons per milliliter. That is significantly better than what we see in the marketplace from other lateral flow tests. These are easily scaled. As you can see, some pictures that are there, they're based upon standard assay cartridges, customized lateral flow readers. What you see there is a QIAGEN reader, and proven commercial assay platforms. We have commercialized these for tropical diseases in Africa and Asia, and we have ongoing trials for tuberculosis and for cytokine trials in Europe. In all cases, the results are achieved in less than 15 minutes. We couldn't do this alone. We're a small company. Maven leverages a large and strong collaboration network, primarily in New Jersey. You see many of the organizations listed in the center there. They're conducting clinical trials and helping us to achieve regulatory approval. This new test platform should open up a number of use cases, and they're shown in the circles there. One I want to highlight, and it was mentioned earlier, is looking at cytokine storms or cytokine imbalance. We have a significant amount of experience showing that we can identify IL-6 at very low levels, which is important because it's being seen that IL-6, which then leads to inflammation, comes up very quickly in CoV-2 and usually has a much higher pronouncement in CoV-2 patients. Finding that early might allow us to do earlier treatments for patients before they get to a cytokine storm. In all these cases, we expect to have EUA approval for the high complexity antigen test in July, followed by nasal swab, saliva, and then even we're working on pre-symptomatic surveillance tests. In conclusion, at Maven, we're proud to be working alongside such a wide range of colleagues in New Jersey, and we hope that our small part can help out everybody that's really suffering from this disease. Thank you.
Thank you so much. Thank you, Richard. And now our BioNJ Vice Chairman and the CEO for OncoSec Medical, Dan O'Connor. Welcome, Dan.
Thanks, Debbie, and thank you very much for allowing OncoSec to present during this forum today. It's particularly encouraging to see the work that Phyllis has shown us that's happening amongst all the companies around the world to gather together and as our last speaker just said, solve this disease and solve for this disease. So really proud to be part of that effort as well. OncoSec's a cancer immunotherapy company. That's our origin. We've been taking intratumoral IL-12 and electroporating that into tumors of cancer patients with multiple different types of solid tumor cancers. We've shown monotherapy activity when we do that. In other words, our drug on its own is able to shrink tumors that are treated and untreated, including visceral lesions. So we've shown an abscopal effect as a monotherapy. And when we combine with Keytruda and anti-PD-1 checkpoint therapies, we've shown an ability to take patients who've not gotten a benefit from checkpoint therapy and give them IL-12 intratumoral and see a response to the checkpoint therapy. So that's yielded partnerships with Merck and others for the company, and that's our core business. When this pandemic first occurred, we looked at ourselves as cancer researchers and said, like many others on this call, and said, what can we do to help solve this problem? And in that connection, we realized that IL-12 has the ability to be obviously a very strong pro-inflammatory signaling cytokine and could potentially increase the efficacy of a vaccination or a vaccine approach. So like all scientists do, we wrote our ideas down on a paper, a white paper actually, shared that with the NIH and Providence Health. When shared with the NIH, they appreciated the idea of using IL-12 as an enhancement and offered to license to the company the spike protein that they've developed. So that occurred. They sent us their spike protein, again, sharing the same concept with Providence Health. They also appreciated the concept of using IL-12. The researchers at Providence Health are cancer immunologists like the folks at OncoSec. And in fact, the cancer doctors there as well really realized that the application of their understanding of cancer immunotherapy could be translated into infectious disease setting. And so they offered to file an investigator-initiated IND, and that IND has been filed. Preclinical research is ongoing currently both at Providence Health and at OncoSec. The study design for Providence's investigator-initiated IND is a 2-arm study, approximately 40 patients, looking at NIH's spike protein using electroporation, which I should say is an established vaccine delivery platform. So using NIH's spike protein delivered by EP alone, and then looking at that compared to the addition of IL-12. So that's the design of the program. We're also using that same design preclinically and looking forward to moving this program forward. So again, very much appreciate the opportunity to be here. Encouraged by the number of vaccine programs that are in the pipeline, as well as all the other programs that are coming forward. But I would remind us all that we need multiple shots on goal. You know, the hit rate for vaccine studies is relatively low. So we're going to need these programs in the pipeline to increase the opportunity for us to see an efficacious vaccine come forward as quickly as possible. Again, Debbie, thank you so much for including us. Phyllis, thank you as well for all that informative information at the front of the program.
Thank you so much, Dan. Our pleasure. And now the President and CEO for PharmaJet, Chris Capello. Welcome, Chris.
So PharmaJet is a needle-free injection device commercial stage company where we've established 2 base needle-free injection platforms shown here on this slide. One is for intradermal delivery and the other is for intramuscular subcutaneous delivery. We're the only commercialized alternative with FDA clearance for vaccine injections other than needle and syringe. We're also the first and only needle-free injection technology that's received WHO pre-qualification. The technology has been proven to be safe, fast, effective. For example, we have a long established relationship with the WHO around polio eradication, where recently we participated in a large-scale campaign in Karachi, Pakistan, where very similar to what we're doing today, we trained 50 caregivers over video WebEx in Karachi, and they went out and subsequently trained 2,200 caregivers on our technology. And in 5 days, they vaccinated over 500,000 children for polio. And this really validated the utility of the devices, as well as demonstrated the ability to increase vaccination rates in this campaign by 18% for those groups that were using the needle-free devices. So this type of success with the technology has really spurred important collaborations and pharmaceutical development partnerships. PharmaJet has over 50 active collaboration partners working on novel pharmaceuticals. Some of those indications span from novel oncology to SARS-CoV-2. And on that thread, our device is currently in over a dozen different SARS-CoV-2 vaccine development programs, 7 of which are in testing already, and we hope to move those from preclinical to phase 1 here this summer. The technology has demonstrated its ability in some cases to actually make vaccines work better compared to needle and syringe, and it's allowed many of our partners to move away from maybe archaic or older technologies such as electroporation and really move towards new needle-free delivery technologies. We're also actively working with the federal government to support additional domestic manufacturing capacity to support these vaccines. As Oncosec said, we need many, many shots on goal, and we hope to be a part of that when they become available. This is the update from PharmaJet, and we would welcome any discussion around collaboration. Thank you for your time today.
Thank you so much, Chris. And now to the founder and CEO of RTM Vital Signs, Nancy DiGianni. Welcome, Nancy.
As we've seen over the last few months, COVID-19 is killing people, most of them in very vulnerable populations who would truly benefit by having a hands-free, objective way of detecting a worsening lung function caused by COVID-19. So why is this important? Because the progression of respiratory distress can occur very quickly, even over a period of a few hours. And because some of the people infected don't have a fever or experience any breathing difficulties until COVID is well advanced with permanent lung and organ damage or death. Many of our most vulnerable are those that are least able to assess their own health situations, including those in nursing homes and the elderly who live alone. There are some wearable monitors available today which can determine respiratory rate, but that's only half the issue. Tidal volume, or the volume of air that we breathe, is the other half of the respiratory function, and the RTM monitor can determine both. We already have a prototype device which we've been developing for opioid overdose prevention and which can be modified for early detection of COVID-19. Our monitor has already been designated by the FDA as a breakthrough device. It's wearable, it's wireless, and when coupled with our diagnostic algorithms, it can accurately detect and predict the progression of respiratory depression. In addition to continuously monitoring the respiratory rate and the tidal volume, our device will soon also include measurements of heart rate, temperature, and oxygen saturation. And these are the same key vital signs that are needed for early detection of COVID-19. The measurements will be continuously analyzed and converted into a risk index, displayed on an individual smartphone, or automatically sent to a physician or other caregiver with an alert or an alarm of worsening COVID-19 infection. With the RTM respiratory monitor, timely decisions can be made whether a patient is well enough and stable enough to stay at home, should see a doctor, or be in a hospital. In short, early detection of COVID-19 can save lives. Thanks for this opportunity this morning, and please visit our website and contact us for more information. Thank you all.
Terrific. Thank you so much, Nancy. And now welcome to the Chief Scientific Officer of Sologenics, Dr. Oriola Danini. Welcome, Oriola.
Good morning, everyone. Thanks for having me. Sologenics is based in the Princeton, New Jersey area and develops products to address unmet medical need across both our business segments. In our specialized biotherapeutics segment, we have 2 advanced programs in oncology and oncology supportive care, while in our public health solutions segment, we have both drug and vaccine efforts. In public health, we are leveraging our existing thermal stabilized vaccine platform for COVID-19, and this has grown out of our very productive work with the University of Hawaii at Manoa. So there are 3 keys to our vaccine platform. Firstly, we use a protein platform format. This is really important because the protein format is the gold standard for clinical safety. So this means we can address the broadest possible population, including those that are immunocompromised or elderly. We also carefully select the adjuvant so that we get the maximal potency possible for our vaccine. In this case, we have a novel adjuvant that's been clinically tested and which we've shown elicits a Th1 response, which is believed to be very important for COVID-19. And finally, we have a thermal stabilization platform. This is really important because it allows us to maximize our distribution and storage by producing a vaccine that can be shipped ambiently and is actually, for some of our vaccines, has been shown to be stable to temperatures as high as 40 degrees Celsius or 104 degrees Fahrenheit for at least a period of a year. So what does this mean specifically for COVID-19? Well, firstly, the antigen of interest in COVID-19 is the spike glycoprotein. So this is a trimeric glycoprotein that's on the surface of the virus itself, enabling easy recognition. However, producing multimeric proteins and those with glycosylation can be a bit challenging. And we're using an S2 cell expression system, which we've produced multimeric glycoproteins before, specifically in the context of Ebola. We're also using, as I mentioned, the co-vaccine adjuvant. This is really important because not only do we stimulate antibody responses, but we also stimulate cell-mediated immunity and with the Th1 response, as I noted. Preliminary experiments suggest a really strong response after only a single dose of the vaccine. And finally, as I said, we're focused on thermal stabilization because at the end of the day, when you want broad distribution, it's really important to have a clinically convenient product. So here what we're looking for is individual vials. They've been thermal stabilized. They can be shipped at ambient temperatures and they can just have water added at the last minute prior to injection. So very simple to use. So we're very excited about our approach as we go forward, and we look forward to further successes. Thank you.
Terrific. Thanks so much, Oriola. Appreciate it. Thank you to all of our presenters. So now we have time for a few questions from the audience, and we do have a few here. So let's see. So question for Phyllis. Great information and thank you. A little off topic from R&D for COVID, but is Bio seeing any supply chain issues, specifically API and the concentration in China?
So a great question. Actually, we've been looking at this, and most of the products that our companies manufacture don't require chemical supplies, so they're not exactly suffering from API issues. Many of our antibiotics companies did actually, when we started to first see the outbreak, actually stockpile API and have told us that they are not seeing supply chain issues at the moment. But this is a very important problem, and we're making sure to work with the federal government on new strategies for ensuring that we have redundancies in supply of not just API, but other raw materials that are important for making biologics and drugs.
Terrific, thank you. And also, so this isn't directed to anyone, but Phyllis, if you'd like to take it, terrific. And if someone who's expert on this topic on the line, we'd welcome your comments. So how often would you expect that recombinant human plasma gelsolin, will need to be given? And what would you see as primary parameter to measure success, clinical labs, et cetera?
So I think that's one of the companies, one of the gentlemen talked about the human plasma product. So I will defer to that CEO.
So maybe in the meantime, we'll come back to that one, but in the meantime.
Hello.
Oh,
hi.
We're there? Okay, great.
Yes, it's me, Susan. Sorry, I couldn't unmute for a minute there. Could you just read the question again? I was busy trying to unmute as you read it.
Sure. How often would you expect that recombinant human plasma gelsolin will need to be given? And what would you see as primary parameters to measure success, clinical labs, et cetera?
So that's a fairly complex question. The current plan for our upcoming trial is 3 doses in the hospital, intravenously, to patients, and we have a whole host of both primary and secondary outcomes, including status 14 days, 30 days, 60 days, and 90 days for those patients. So We're looking at organ function, mortality, and some combination endpoints. But I'd certainly be happy to provide the full protocol for the person who asked that question after the session.
Terrific. And all of our presenters' contact information will be included. These slides will be included on the link at the BioNJ website tomorrow, so you will be able to contact. So that was Dr. Susan Levinson of BioAegis Therapeutics. Thank you, Susan.
Thank you.
Yes. So Phyllis, timing on vaccine, you know, we hear lots of stories. What are your thoughts?
So great question. It's probably my most frequently asked question. So our understanding is that there are at least a handful of vaccines going into clinical trials, more advanced clinical trials this summer and into the fall. As you've heard, the US government has a goal of having at least some doses available between October of this year, which is very aggressive, and the first quarter of next year. And when I talk to the vaccine companies, they say that is feasible. Companies are actually starting to manufacture doses at risk while they are doing the phase 2 and 3 trials. So that is definitely unprecedented. If indeed their vaccines are shown to be efficacious, at least in some limited or a reasonable amount of efficacy, those doses will be produced and made available, and the CDC will start to allocate them across the most at-risk populations. So it's feasible and possible. Of course, the clinical trial work is the most important part.
Great. Thank you, Phyllis. Thank you.
Sure.
So question, is anyone seeing your COVID-19 clinical trials impacted in a positive way versus perhaps other trials that you've done in the past?
I guess I would just say that the one benefit, unfortunately for patients, is that the recruitment is very fast for clinical trials because there are so many patients. So if that's a silver lining for testing all these products, I think that's important.
Terrific point. Thank you, Susan. Anyone else want to comment on that one? Okay. Phyllis, question for you. I know, actually, I'm just in awe of the fact that you're with us today because you have a little meeting going on next week there at Bio. And I know that you personally have been, had a really instrumental part in one of the featured components. So can you tell us a little bit about what you've been working on and what it might look like next week, please?
Absolutely. We're very excited. We have quite a fantastic lineup of COVID-19 sessions, as well as a full complement of all the other great sessions we always do at the convention. Interesting to take a 16,000 person meeting and put it into a digital format. So it'll be interesting to see how this goes. On Monday, we have the opening remarks by our new CEO, as well as our immediate past CEO and our great chairman of the board, Jeremy Levin. And then I'm actually moderating a panel on therapeutics being developed for COVID-19. That's one of the live sessions. And we have 5 great CEOs and leaders who are working on different kinds of therapeutics. And then Tuesday is sort of the big day of plenary. We have a one-on-one interview between our new CEO, Michelle McMurray-Heath, and Dr. Anthony Fauci. That should be very fun. And then we have 2 panels on vaccines, one on the clinical issues of developing vaccines that features Dr. Peter Marks, several different companies, heads of R&D, including Paul Stoffels from J&J, and Dr. Francis Collins will be on that one. And then the second one is actually on implementation, which I think we all agree globally is going to be the second Herculean thing we do. After making a whole bunch, we have to give it to millions of people, hopefully relatively at the same time. And we have someone from CEPI, someone from Gavi. We have Monsef Salawi, who's running Operation Warp Speed. So we have some really great people who will be talking about that last couple of miles from the great development to giving the vaccine. There's also a great session on the ethics of doing clinical trials in a pandemic that happens later in the week that will focus on some of the work being done and how you do clinical trials in an outbreak setting, which I think is one of the important scientific and ethical issues we should talk about. And then there's a lot of pre-recorded content that I would call your attention to. Some great sessions from the DoD on their work in terms of rapid response, and a great session from BARDA on the work they're doing and working with companies on blood products, which is one of the most important things they have to do in preparing for responding to any kind of pandemic we may face. So a lot of great information. Get into the site and fish around, and then know there's a COVID-only specific track for company presentations. So very much like what you just saw today, companies will have just a little bit longer, but not much longer, a little bit longer to present their COVID-19 research for partnering and in order for investors and partners to see it. So there's COVID-19 content and then the rest. Thanks for asking, Debbie.
Yeah, no, terrific. I can't wait to see it. You know, frankly, as an organization that does so much of our work is, you know, reliant on bringing people together at meetings. You know, figuring we're going to have the best example possible of how to do it next week. So we're very much excited to that.
And let me add, if you are the companies I heard, the presentations were amazing. I really am glad I stayed. A lot of great work going on. If you have not signed up to do a COVID-19 company presentation at the meeting, know that for companies working on COVID-19 specific products, you can actually register for the meeting for free and do partnering. So if you haven't done that already, either let Debbie or I know, and we can connect you to the registration code for companies that want to specifically focus or have COVID-19 products and want to specifically present their company's work in the COVID-19 company presentation track. So I want to make sure that everyone here gets to present again to a broader global audience.
Terrific. And maybe they'd have a little more than 3 minutes.
They will have more than 3 minutes, although you guys did great in 3 minutes, but you'll have more than 3 minutes.
The format worked, I think, hopefully. Thank you for that. That's a tremendous service that you're providing at Bio. We have one more question, and then we'll move to close. So how do you balance the work internally between current programs and the new programs? Do you bring in additional resources or rely heavily on your CRO? Question, we have one person who wants to take a crack at that one.
Yeah, Debbie, I think maybe I could just give a quick comment. I think, you know, I was appreciated the fact that when we shared this idea that we had one of the top hospital networks in the United States, not-for-profit hospital networks in the United States, Providence Health, saying that they wanted to conduct this clinical study and that they were willing to fund it, which was tremendously helpful. So, you know, that I think again is a testament to everything we've been seeing. You know, the amazing evidence of the human spirit coming together. But I guess I would encourage everybody who's got a concept and an idea, once you distill it to, you know, a writing and, you know, some evidence and some justification around it, share it, because you never know. You might get somebody who's like Providence Health, who appreciates the idea and is going to want to, you know, move it forward, you know, with their own resources.
Terrific, Dan. Thank you. That great point. And also, if BioNJ can be a conduit to that, and clearly you've heard that Phyllis, Arthur, and Bio are more than willing to help share your story. We would be thrilled to have that, to play that role at this important time. So I would like to thank all of our presenters for the work that you're doing on behalf of patients. Clearly, thank you, Phyllis, so much for what you're doing at Bio. It's so important and so inspiring too, I have to say. A reminder that the recording of the webinar, along with the slides, will be available on the BioNJ website tomorrow, bionj.org. Contact information for each of the presenting companies is included in the slide deck. Thank you again to today's sponsors, our supporting partners. If you're interested in learning more about BioNJ, our team member, Kim Minton, would be very happy to tell you any of our partners. Also, we hope that you'll join us again on Tuesday, June 16th from 10 to 11, where we will do this again, although this time it will be with 15 companies. And we'll also hear from Dr. Bob Harari, who has two cell therapies addressing COVID. So that should be an interesting conversation. So everyone, please stay safe and well. We need you. Our patients need you. And we look forward to seeing you in person again sometime very, very soon. Thank you again. Have a great day.